leader of group 4. I'm Celine, and those are my teammates.
Hi, my name is Yi Xiong Mi.
And then,
Hi, my name is Muhammad Azwan.
Hi, my name is Tan Jing Yi.
so last, my name is Xiao Ho Yi. Okay.
Okay, the first question is we need to find 8 words from the crossword table. After find all the 8 words, we need to choose any 4 words. After that, we have to use all the alphabet in that words to form a peptide model, and I will explain it later. Okay, the first word that we choose is the word C. So the S stand for serine, and E, the middle one, stand for glutamic acid, and the last one, D, stand for aspartic acid. Okay, the second word that we choose is word mid. The M stand for methionine. The alphabet E in the middle stand for glutamic acid. However, the alphabet T stand for threonine. Okay, the third word that we choose is test. The alphabet T at the end stand for threonine. Alphabet E stand for glutamic acid, and alphabet S stand for serine. Okay, the last word that we choose is word nest. The alphabet N stand for asparagine. Word E stand for glutamic acid. S stand for serine, and the last alphabet T stand for threonine.
Now is our question 1, part B. We need to build a model that containing alphabet C, A, C, I, N, G. And here is our C, A, C. Here is our possible disulfide bridge.
Continue with the I, N, and G. Here is the C terminal because the free carbonyl group is located at the right. Okay, next, here is our Chachin model, reverse order, which is here is G, here is N, here is I, C, A, and C.
We also found that the disulfide bridge is here, and the difference in the two model, that is the C terminal is on the capital letter C, whereas it's also got the free carboxyl group. In conclusion, that the sequence will affect the C terminal, and we know that the C terminal is always located in the right hand side.
For question 2, the question needs us to choose 3 types of the lipids. One of the lipids that we choose is phospholipid. Phospholipid divided by 2 parts, which is polar group, which also is the hydrophilic head, and the non-polar group is the hydrophobic tail. For the polar group contains the alk group, phosphate group, and the glycerol group. For the non-polar group divided by 2 parts, which is saturated fatty acid and unsaturated fatty acid. To determine which is the unsaturated fatty acid, we can see for the CC double bond.
Since our long fatty acid these two long fatty acid chains are non-polar, hence we know that it will avoid water due to their insolubility. As a result, we can conclude that this phospholipid exhibits selective permeability, which means that it controls the movement of substance in and out of the cell. Okay, so for the second model that we need based on the question 2 is sphingolipid. So normally sphingolipid are found in all animals, plants, and fungi. Sphingolipid are formed through the union of a very long chain fatty acid with sphingosine. As you can see, there's a amide bond which link the fatty acid chain and the sphingosine together. So sphingolipid function as structural component of membrane, lipoprotein, skin, and others biomaterial as well. It also also function as the signaling module. The last model for the question 2 Roman 1 is cholesterol. It consists of 3 member rings and 1 5-member rings that all fuse together. Cholesterol is a principal component of animal cell plasma membrane, and typically there's a smaller amount of cholesterol are found in the membranes of intracellular organelles. The relatively rigid fused ring system of cholesterol and the weakly polar alcohol group at the C3 position, which located at here, have the important consequences for the properties of plasma membrane. Not only that, cholesterol is also a component of lipoprotein complex in the blood as well.
This is the triacylglycerol, and this part is the glycerol, and this is the ester bond, and this three is the 3 fatty acids. And triacylglycerol are rich in highly reduced carbon, and thus it can yield a large amount of energy in metabolism. And the glycerol part does not mix with water and break down, so it can store energy. And triacylglycerol is not a component of plasma membrane because it does not have the hydrophilic part like phospholipid, so it could not form the lipid bilayer with the plasma membrane. Okay.
Okay. The similarity between of RNA and DNA is both oligonucleotide contain a pentose sugar, phosphate group, and nitrogen base.
For the differences between the DNA and RNA is, first is the pentose sugar. The DNA got 1 hydroxyl group only, and for the RNA got 2 hydroxyl group. The second differences between the DNA and RNA is the nitrogen base. For DNA, nitrogen base is the thymine, and for the RNA is the uracil. That's all.
Question 4 revealed that the two anomers here, which is here is the metal alpha D glucoside, and here is metal beta D glucoside, and the difference between the alpha and beta is which the CH3 here for alpha is on the bottom and for the beta is on the top. These two anomers show there's no positive result in tolerance reagent because it's not reducing sugar since that it have no free aldehyde group. For question 4 part 2, the monomer within carbohydrates will show positive result for tolerant reagent that we choose is glucose. Glucose will form two forms, which is first is alpha D-glucose and second is beta D-glucose. The alpha D-glucose is determined by the OH group on the bottom, while the OH group of beta D-glucose is on the top. More that the glucose will show multi-rotation structure, which is multi-rotation is meant by the observed change in the optical rotation of the alpha and beta anomers of glucose when dissolved in water. Due to the ring change tautomerism, the alpha and beta form slowly in the converts until equilibrium is established. The alpha anomer of D-glucose has a specific rotation of positive 112 degrees in water, while the beta anomers of D-glucose has a specific rotation of positive 19 degrees. Well, either anomer is dissolved in water, the value of the specific rotations change over time, eventually reaching the same value of positive 52.5 degrees.